In this chapter, we'll be studying microbial genetics. Genetics is the study of the heredity of living things. Depending on how much you remember from your general biology course, this chapter could be predominantly review for you, but I bet you'll find some new nuggets of information. We'll be exploring DNA, its structure, the formation of proteins, and the other products associated with them. Here's some quick fun facts about DNA. As you recall, the DNA is the information of the cell. It's the instructions for anything that the cell is. The complete cell's DNA sequence and all its inclusions are called the genome. So naturally, a bacterial genome is much smaller than a human genome. There's about 0.4 to 9.4 million nucleotides in each individual bacterial cell. And the human genome has about 4 billion nucleotides, so it's much bigger. It's 1,000 times larger than the average E. coli, and it only has about 6 times more genes, so about 24,000 in humans versus 4,000 that we see in E. coli. What that results in is 85% of the genes in an E. coli actually code for proteins, whereas only about 2% of our genome codes for proteins. So probably the actual gene number is not that different. However, the human genome contains a lot of other stuff, which until recently we thought was just junk. However, it turns out that this other stuff probably regulates a lot about gene expression and thus changes the ways in which the human genes are expressed. Anyway, let's focus on the microbes for now. So genetics is the study of inheritance or heredity of living things. It involves the transmission of traits from parents to offspring, the expression and variation of those traits, the structure and function of the genetic material, and how this material changes or evolves. It takes place on several different levels: the organismal level, the chromosomal level, and the molecular level. When we consider the organismal level, we're considering the whole organism or cell. At the chromosomal level, we're examining the characteristics and actions of the chromosomes. And at the molecular level, we're dealing with the biochemistry of the genes themselves. All of these levels are useful areas of exploration. But in order to understand the expressions of microbial structure, physiology, mutations, and pathogenicity, we need to examine the operation of genes at the cellular and molecular levels. So that'll be the focus of this chapter. So for survival, a species has to have the capacity to first be able to self-replicate. In single-celled microorganisms, this means division of the cell by binary fission or budding. But really, it's much more than just cell division, because we have to have accurate duplication and separation of DNA, their genetic material, which in most microbes is one long circular molecule of DNA. Now, when we consider a genome, we're considering the whole complement of genetic information inside a particular organism. So that's not only the DNA in the chromosome, but any other satellite DNA, let's say. In microbes, you can see that we have one single circular chromosome. In many bacteria and some fungi, we'll also see plasmids, these other small, tiny circular pieces of DNA. In a eukaryotic cell, there are also organelles that contain their own DNA. Do you recall which those are? We have both chloroplasts as well as mitochondria. Extrachromosomal DNA components are all part of the organism's genome. So the genomes of individual organisms are unique. However, the pattern of nucleic acid structure and function is similar among all living organisms. And if you think about that, that's amazing that since the beginning of time, nucleic acid structure has remained consistent. A chromosome is a discrete cellular structure which is composed of neatly packaged DNA molecule. Now, in eukaryotics, we have numerous linear chromosomes. However, in prokaryotes, again, the usual arrangement is a single circular chromosome. However, on occasion, we will see sometimes they have a linear chromosome or multiple circular chromosomes. But in general, a prokaryotic cell has one single circular chromosome. Now, what exactly is a gene? There are multiple definitions that we've had throughout time, but the current one we'll stick with is that a gene is a segment of DNA that contains the necessary code to make up a protein or a group of related proteins and RNA molecules. There are three basic types of genes. There are structural genes, which are the genes that code for physical proteins that manifest in the phenotype of the organism. There are genes that are for the RNA machinery that's involved in producing these genes. And then there are regulatory genes that code for proteins that regulate the process of gene expression. Now, a quick review of genotype versus phenotype. Probably you'll recall that in humans, the phenotype is what we see, and the genotype is the genetic information behind that. Now, it's the same for our microbes also. But the genes, instead of coding for things like hair color or eye color as a phenotype, will code for things like capsid structure or the presence or absence of a flagella. So genotype, what the gene actually says, and phenotype, what it actually looks like, how it manifests itself. So phenotype for us might be eye color and hair color. Phenotype in a microbe would be whether it has the flagella or what its capsid looks like. Now, genomes vary greatly in size. Small viruses might have only 4 or 5 genes. A bacterium like Escherichia coli has about 4,288 genes. And a human cell could express anywhere from 20,000 to 25,000 different genes. If we stretched out the genes in an E. coli, it would be about 1,000 times larger than the cell itself. It might be a millimeter when the cell itself is only a micron. If we unraveled all the DNA in a human cell, it would stretch out about 6 feet long. And however does all of this fit inside a cell? All it comes down to is how these chromosomes are coiled and packed. So it isn't like just one long, skinny piece of yarn. It's associated with proteins and wound up very tightly so that it fits into the cell. It takes up only about 1/3 to 1/2 of a microbial cell's volume. Examining DNA at the molecular level is going to require us to take a really close look at its structure. To do this, let's imagine that we're able to magnify a small piece of a gene about 5 million times. A little bit on the history of the structure of DNA and how we discovered it. It was elucidated in 1953 by James Watson and Francis Crick. At least, those are the guys that get the credit for it. There was another critical person involved who actually produced the images that allowed them to understand the double helical nature. Her name was Rosalind Franklin. Watson and Crick get the credit. They discovered the basic structure of DNA, that it's made of nucleic acids, and that there are 2 strands that combine to form the double helix. This is the famous picture of their huge structure of DNA. You'll probably recall that the basic unit of DNA structure is the nucleotide. The nucleotide is composed of a phosphate, a deoxyribose sugar, and a nitrogenous base. So at the center, I have my deoxyribose sugar. It has a phosphate attached to it and a nitrogenous base like adenine, guanine, cytosine, or thymine. Another critical component of this sugar is the 3' OH group. Here we can see the same structure represented, where we have a sugar, a phosphate, and the nitrogenous base. Each nucleotide is covalently bonded to the next nucleotide down in the strand in order to form a single-stranded DNA molecule, and then each of those strands are hydrogen bonded to each other in order to form the ladder-like structure. So again, here we have one nucleotide with a phosphate, sugar, and nitrogenous base. Each of these nucleotides is covalently bonded to the next nucleotide down in the strand. So here's our next one. In this way, we build one long single strand of DNA. This strand of DNA is linked to a complementary strand of DNA over here, and the bonds that you see down the center are hydrogen bonds. So the bonds holding the two strands together are weaker than the bonds that hold the nucleotides together. And this makes sense because if we needed to unzip the molecule, let's say to replicate it or transcribe a gene, then we can break it apart down the center easily and access the code that's written on the nitrogenous bases of each nucleotide. You'll also probably recall that adenine and thymine always pair together, and guanine and cytosine always pair together. So I remember it like this: G and C, because those are my initials, George Cornwall. And T and A, well, you can work on that one. I mean teaching assistant. You may also recall the anti-parallel arrangement, where we have one DNA strand that's in the 3' 5' orientation and one that's in the 5' 3'. That's where that 3' OH was really important. It's this bond here between the 3' OH of the sugar and the phosphate. So the phosphate of the next nucleotide is bound to the 3' carbon or 3' OH group on that sugar molecule. So what that lends itself to is us having a 3' OH group at this end and a phosphate attached to the 5' carbon. So thus we have a 3' 5' orientation. On the complementary DNA strand, we'll see that the 3' OH hangs out on this end, and the 5' phosphate group is hanging out on this end. So in that sense, the molecule is oriented in an anti-parallel direction. One more component that we might consider are these nitrogenous bases. We have cytosine, guanine, thymine, and adenine in a DNA molecule. Two of them have a double ring structure, whereas the other two have a single ring structure. The double ring structure are purines, which are adenine and guanine. And the pyrimidines are a single ring structure, that is T and C. You'll notice that always a purine is paired with a pyrimidine. And in order to keep these straight, I have a couple of little rules. Purine and pyrimidine are different length words. The big word goes with the single ring structure or the smaller structure. So pyrimidines are smaller or single ring. And the smaller word goes with the bigger structure. So purine, being a smaller word, goes with the double ring structure. The other thing I use is the rule of Y. How do you remember which ones are pyrimidines and which ones are purines? Well, the pyrimidines both have a Y in them. So the rule of Y, PY rimidines is CY tocine and the Y amine, right? So all the rule of Ys, large structure, small structure, small word, big word, opposite. Now let's take a look at a nice animation of DNA structure.
The structure of the DNA molecule was first inferred by James Watson and Francis Crick, based primarily on X-ray crystallography data collected by Maurice Wilkins and Rosalind Franklin, and chemical analysis of base composition of DNA conducted by Erwin Chargaff. The key features of the structure are its right-handed double helical structure. Each helix consists of an alternating sugar-phosphate backbone with nitrogen bases projecting toward the interior of each helix. One complete 360-degree turn of the helix covers 10 bases of length and equals 3.4 nanometers in physical distance along the axis of the molecule. The width of the double helix is 2 nanometers. The nucleotide bases are attached inside each backbone of the molecule so that the nucleotides in one helix or strand are hydrogen bonded to the bases in the other helix or strand. The hydrogen bonds hold the two strands of the double helix together. An adenine in one strand is always paired with a thymine in the other strand, and a guanine in one strand is always paired with a cytosine in the other strand. Note that guanine-cytosine base pairs form 3 hydrogen bonds, while adenine-thymine base pairs form 2 hydrogen bonds. This makes guanine-cytosine base pairs more stable than adenine-thymine base pairs. Nucleotide pairing between strands also allows the sequence in one strand to determine the sequence in the complementary strand. The two ends of a strand are not identical. Note that on one end of each strand, a 3' hydroxyl group of the deoxyribose sugar is not involved in the backbone, or we say it's free, while at the other end of the same strand, the 5' hydroxyl group of the deoxyribose sugar at the end is free or may contain a phosphate that is free and not bonded to another deoxyribose sugar. This dissimilarity of the two ends of a strand creates the ability to uniquely distinguish each end of the strand. Because of this polarity of each strand, we can see that the two strands of DNA are oriented in opposite directions, or we say that they are antiparallel. In three-dimensional space, the two helices are not equally spaced along the axis of the molecule, such that a major groove and a minor groove are created by the asymmetric spacing of the antiparallel helix backbones. The major groove and minor groove play important roles in the binding of proteins that regulate gene transcription.
Now a quick concept check. Which of the following is not a pyrimidine? Well, that one is pretty straightforward because we learned what? The rule of Y's. Both cytosine and thymine have a Y, so adenine is the only one that is not in the pyrimidines.